Hello everyone, I'm Bibek, a second year Masters in Physics student at the Department of Physics, and my topic of presentation is study of cellular respiration in high pressure. So pressure is a well-known thermodynamic variable, and it has been found that pressure significantly affects the cell metabolism. So imagine a cell in an ocean, and if it goes to certain depth, then several things might happen. It might lose the motility, the ability to find the food by this cell could get reduced. Growth and DNA replication also could be inhibited. And if it somehow goes to the bottom of ocean, it might even die. But even in such harsh environment, scientists have found that microorganisms can survive such extreme environment. So there is still need to understand how these organisms survive in such extreme environment. So cellular respiration is a complex process that occurs in cytoplasm and in mitochondria, and most important part of the cellular respiration occurs in the inner membrane of the mitochondria, also known as the electron transport chain. So one of the important cofactor that takes part in the cellular respiration is NADH. So NADH in its reduced form is fluorescent in nature. So it turns out that we can monitor the cellular respiration with the help of the autofluorescence coming out of the NADH when we excite it with UV pulse. So for our sample, we use Saccharomyces cerevisiae, also known as Baker's yeast, grown on YPD plates for 2 to 3 days. We triple wash our sample and load it to a special chamber known as pressure chamber, as you can see in the second photo. So if you looked closely into the pressure chamber, it looks something like this, and our sample goes inside it. It is trapped there. And we excite our sample in the optically viable region of the pressure chamber. So spectroscopic setups look something like this. We have a nitrogen discharge laser that can emit a ultraviolet pulse of about 337 nanometer, which excites our sample. So what we do is we excite our sample with this excitation pulse, and in response to that, our sample emits back an emission pulse. And we take a slice of time in that emission pulse, which gives us the spectrum, and this spectrum is then sent to the computer. So this process is also known as autofluorescence. Autofluorescence is the natural emission of light by biological molecules when they have absorbed light. So in reality, our optical setup looks something like this. So to simulate the high pressure environment, we have developed a system also known as high pressure perfusion system, and this system contains several two pressure generators, several valves, pressure tubings, and fluid reservoirs. So it can provide us the pressure ranging from 0 to 700 atmospheric pressure. So in reality, high pressure perfusion system looks something like this. So in total, the high pressure perfusion system, or also called microperfusion system, works in conjunction with a spectroscopic setup for our experiment. So we use a special method known as spectral phasor analysis to analyze our data. So it turns out that a spectrum in a spectral phasor is just a dot, and it has two parts, the real and the imaginary part. So spectral phasor analysis has graphical and analytical interpretation. So looking at the shift in the spectral phasor, we can predict several things that's happening in the cellular respiration. So one of the important observations was ethanol inhibition in high pressure. Ethanol is a well-known cellular respiratory inhibitor, and it has been found that it affects the biological membrane. So it is important to study this inhibition because most of the cellular respiration occurs in the inner membrane of the mitochondria. So the spectrum looks something like this, and the intensity profile looks something like this. So our intensity was decreasing prior to the perturbation, and after the perturbation at about 20 minutes at the ambient pressure, the intensity increased. And similar observation was found in the high pressure. So looking at the spectral shift, we see that the spectral shift were in opposite direction at 0 bar and 300 atmospheric pressure. And that tells us that the effect of ethanol at high pressure to the membrane is opposite to the effect of ethanol to the membrane at low pressure. So another observation that we looked at was cyanide inhibition at high pressure. So cyanide is also well-known respiratory inhibitor, which is found to affect the complex 4 of the electron transport chain. So looking at the cyanide response, the spectrum looks something like this, and the intensity profile looks something like this. So the intensity was somehow stabilized, somehow stabilized, although it was dropping. There was some loss, and as soon as the perturbation was carried out, the intensity increased with significant amount. And if we look at the spectral shift at the ambient pressure and the high pressure are towards the same direction. So that is the strong evidence that the response due to cyanide at a high pressure of 320 bar and the ambient pressure is similar. So what we can say is that cellular respiration was still occurring at 320 atmospheric pressure. So in conclusion, high pressure perfusion system is a robust way to real-time monitor the cellular respiration in high pressure. So real-time monitoring of cellular metabolism in high pressure is possible using the UV-excited autofluorescence. Ethanol and cyanide inhibition are important tools to investigate pressure effect on the cellular metabolism. So if you have any question, please feel free to ask me. And thank you for watching.